Lastly, I want to address the atmospheric conditions required for bacteria to grow. Bacteria will either be classified as aerobic, microaerophilic, or anaerobic. The aerobic and microaerophilic bacteria will all grow in a CO2 incubator. And a CO2 incubator is an incubator that has approximately 5% of carbon dioxide injected into it. And that is required specifically for organisms like Haemophilus and Neisseria. They won't grow without that CO2. So we have a CO2 tank that is stored beside the incubator, and that's pumped in continuously. Lastly, we want to address the anaerobic requirements of bacteria. Anaerobic bacteria will die in the presence of oxygen, and so we utilize containers such as these two, an anaerobic jar and an anaerobic box, to create anaerobic conditions to enable anaerobic bacteria to grow. And once we've inoculated these plates with the swab, we'll put them into the container. We'll put an indicator in to tell us whether anaerobic conditions will be met. And under anaerobic conditions, this indicator is white. In the presence of oxygen, it starts to turn pink. And we have a gas pack that's put in, and then the container is sealed. That gas pack produces hydrogen gas, and that hydrogen gas binds with the oxygen in the jar to produce water. So in the morning, there'll be a little bit of condensation on the top of the jar, but all the oxygen inside should be depleted so that anaerobes can grow. As you can see, that white indicator has turned pink, meaning it's been exposed to oxygen, but by tomorrow morning that indicator should be white, telling us the jar is working. These are incubated for 48 hours. For those special cases where anaerobes are specifically requested, we can use a gas pack. It's the same as one of these jars. It's got a smaller indicator and a smaller gas pack. As you can see, the indicator is white, meaning the gas pack has worked, and we can keep these going for 7 days, 2-3 weeks if necessary, to isolate any very slow growing anaerobes. We've talked about culturing bacteria. Now I just want to talk about the gram stain, actually seeing bacteria under the microscope. There's two reasons why we do that. First of all, we do a gram stain in cases where we might get a positive blood culture. The machinist told us the bottle is positive. The doctor wants to know what's in it. It would take at least 24 hours to grow the organism, but we can prepare a gram stain and tell a doctor basically what's there. All bacteria are divided into 4 groups based on gram stain and shape of the organism. All bacteria are either cocci, spherical, or they're elongated, called bacilli, and they'll stain either gram positive or gram negative. So we've got 4 categories: gram positive cocci, gram negative cocci, gram positive bacilli, gram negative bacilli. And in the case of a blood culture or say a spinal fluid, where the doctor has to know if it's a bacterial infection, this gram stain will tell him exactly, well, not exactly, but he'll give him a pretty good idea of what organism is there and therefore what kind of antibiotic to use. If there was a gram-negative bacillus, he'd probably choose ciprofloxacin. If it was a gram-positive coccus, he'd probably use penicillin instead of using a broad-based antibiotic like Rocephin. It just makes it more specific. The other reason we use a gram stain is to examine specimen quality. We're licensed to reject sputum specimens if they have more than 10 squamous epithelial cells per low-powered field. The reason for that is squamous epithelial cells come from the mouth, from the tongue. And the mouth can contain strep pneumo, it can contain Staph aureus, it can contain gram-negative bacilli, all potential pathogens. How is a doctor going to know whether that organism came from the mouth or from deep down? The sputum quality tells them that. If there's a lot of squamous epithelial cells, it's rejected because it's been contaminated by saliva. Also, in cases of wound swabs, if wound swabs have more squamous epithelial cells than white cells, they will be rejected. If they have an equal number of white cells and squamous epithelial cells, we give them the benefit of the doubt and they're accepted. The only exception is burn specimens, because they have to be superficial. There's no other way to culture them, so we make that exception for them. If a specimen is a wound specimen and it's a poor one and it's growing a mixed growth, say of Staph aureus and mixed skin flora, all we have to do, or all we're supposed to do, is simply list the organism. So we would say Staph aureus, mixed skin flora, and no sensitivities. However, if that superficial specimen is poor, but it grows a pure growth, say of Staph aureus, then we'll give it a qualifying comment, but we will list the susceptibilities for Staph aureus and let the doctor decide whether he wants to treat it. Gram stains are done on either swabs or fluids. A swab might be from a surgical patient that you've taken on the floor, and a fluid might be, say, a spinal fluid. With swabs, we take the swab, and after we've cultured it, because you don't want to contaminate it, the plates, we just dab it onto the slide like this. Then we heat fix it till it's dry, and then it's ready for staining. With a fluid, like a spinal fluid, it's centrifuged first, and then we cover it with methanol to fix it. We don't heat fix it, and then we're ready to stain it. There are 4 simple steps to a gram stain. First, we apply the crystal violet stain onto the slide. And that crystal violet stain will stain everything and anything that is on that slide. A little bit too much water. Rinse it off. Take the excess off. The second stage is iodine. Iodine acts like a binding agent or a mordant. And that iodine will bind that crystal violet to the cell wall of all gram-positive organisms, but only the gram-positive organisms. We wash that off. The third stage, and the most critical stage, is to use acetone alcohol to decolorize the slides. We put this on briefly, and this will take off the excess crystal violet that is present. So that at this point in the procedure, all the gram-positive organisms will retain the crystal violet stain. The gram-negative organisms have been decolorized by the acetone alcohol. So for them, we add the final procedure and the final stain, and that's saffron. So what's left on there are gram-negative organisms, and they'll be stained with a saffron stain. That goes on for a few seconds. It's rinsed off. We blot it dry, and it's ready to be examined. I want to talk to you now about MRSA and VRE. First, I'll talk about MRSA. The MRSA plates we use are selective and they're differential. They're selective by using the antibiotic cloxacillin that tells us whether the organism is resistant to methicillin or not. And the agar also employs an enzymatic substrate that is specific to an enzyme found in methicillin-resistant Staph aureus. So if this organism grows on the agar and it's a mauve color, it means it's Staph aureus MRSA, methicillin-resistant Staph aureus. And that's all there is to it. The plate is inoculated and it's incubated 24 hours. If there's no mauve colonies or no growth, the report goes out in 24 hours as methicillin-resistant. And if there's mauve colonies, we know they're methicillin-resistant and that they're MRSA. So MRSA plates are out in 24 hours, whether they're positive or they're negative.